Hello, am I audible? Am I audible to the students? Okay. Okay. Good evening, everyone. This is the last interactive session that we are having, and this is the 12th session, as you all know. So, in this session, we will majorly focus on bioinformatics. So, we will directly go to the first question. So, your first question is: the interdisciplinary field that develops tools, algorithms, and softwares to understand the biology by applying techniques from mathematics, computer science, statistics is known as, and your options are genomics, bioinformatics, cheminformatics, proteomics. Yes, Neha, you are right. It is bioinformatics. So, so what is bioinformatics? Can anybody tell me? So the brief idea is that, but are you aware of what happens in bioinformatics? Have anybody done bioinformatics hands-on? Yeah, yeah, yeah. So basically, you are right. So yeah, in bioinformatics, we have certain tools where we analyze nucleotide sequences, it can be protein sequences. And what do we analyze? We analyze if one nucleotide sequence is similar or how much it is different from another nucleotide sequences, right? So all these are done in bioinformatics. So now for the clarity of everyone, let's go into to check what is bioinformatics. So, as I had mentioned before, bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data. In particularly, these data are large and complex. So, basically, suppose you are analyzing the part of the genome of the bacteria which codes for a particular protein, be it a viral protein, be it a protein for cellular transport. So when you are analyzing it for 1,000 different bacteria, you are taking into account thousands of such data and some 10,000s of such nucleotides for one bacteria. So this actually makes you handle a lot of data, and therefore you require computer to do it, and this work is generally done in silico. So what is it again? So this particular field of science combines biology, chemistry, physics, computer science, information engineering, mathematics, statistics to analyze and interpret biological data. So basically, when we are doing bioinformatics, what we are analyzing, we are basically analyzing biological data, but we are also taking the help of several other fields, be it mathematics, physics, computer science, etc. And as I said, the information is done in silico, that is, it is performed on a computer by a computer simulation, because we need artificial intelligence to do certain kind of these studies. Therefore, computer is required to check the amount of similarity or amount of differences between the nucleotide sequences. Also, it is used to store large amount of data. Okay. And the bioinformatics term was first coined in the year of 1960 by Dutch biologists named Paulen Hoogeweg and Ben Hesper. So according to their research and discoveries, bioinformatics was defined as the study of information process in biotic systems. Now, what does bioinformatics help us do? It helps us to handle huge amount of data and basically not just handle it, but also help us to make a sense of it. So it not only gives us this ability to store a large amount of data, but also analyzes and helps us to do analysis with it, and therefore we can make sense of the data. Okay, so bioinformatics process involves storing an analysis of biological data, and thereby it also further involves creating databases to store experimental data, predicting the way the protein folds up. How do you know do this? Because you will get to know certain nucleotide sequence or certain protein amino acid sequence in with the help of the software tools, and knowing which amino acid forms which amino bonds with which amino acid, you can predict the way in which proteins will fold up in secondary structure, in tertiary structure, or in quaternary structure. Also, it helps us to model all the chemical reaction in the cell and how cells interact with one another. So all these things can be done with the help of bioinformatics, and this therefore is a very broad field which needs diverse range of people with diverse skill sets. So what does this field need? It also needs programmers to write the computer programs to analyze this large volume of data, and also it needs database administrators to organize and store this amount of data. Also, finally, it needs biological scientists which will help to analyze the data, and also web designers to produce sites and apps where the scientist can search all this data. So this is what bioinformatics is. It is basically a potpourri of different kind of informations of nucleotides and protein sequences where we can get them in one place and analyze them. Okay, now let's delve a bit deep into bioinformatics. So in bioinformatics, we deal with different types of data. So the first type of data that we are using in bioinformatics is transcriptomics. Okay, so what is a transcriptome? Can anybody tell me? So transcriptome is basically the set of RNA transcripts in the cell which will code into a functional protein inside the cell. So transcriptomics is the study of that transcriptome, and it basically involves genes which may not be constantly active, and sometimes they can be turned on and off by different kinds of proteins or different kinds of chemical messengers. Now, when a gene is turned on, as you all know, it is basically expressed, and therefore that gene will produce a RNA which will finally manifest itself into a protein. So basically it will get translated, the RNA will get translated and therefore produce a protein. Now in our body, hemoglobin carries oxygen in red blood cells, but that is not needed by white blood cells. So therefore RNA linked to the hemoglobin production in tissues that make red blood cells will not be present in tissues which make white blood cells. So basically the transcriptome of white blood cells will be different from that of red blood cells. So this is how the difference in transcriptome from one cell to another is also studied with the help of transcriptomics. So scientists uses RNA sequencing in computer gene expression, to compare gene expression in different cell types, and thereby in this transcriptomics it can actually compare the transcriptomics of two different cells, be it healthy and diseased, and therefore tell us which one is which one. Okay, so the first step of transcriptomics is RNA extraction. So what happens in this step? In this step, the RNA is extracted from a particular cell, and then it can be RNA or mRNA. Okay, so it is extracted from the cells, and this uh, and during this process, there is a lot of challenges are faced because RNA is not as stable in the environment as compared to the DNA, and therefore it can get easily fragmented by any other external conditions. For example, it can get easily fragmented in the presence of RNAs which are generally present in our fingertips. So therefore we need double autoclaved uh, uh, like all double autoclaved laboratory stuffs in order to extract the RNA, and also we need to maintain sterile conditions during this RNA extraction, because RNA, it is present everywhere in our on our hands, on desk, and even on a piece of pipette. So therefore we have to be very particular during this RNA extraction process. We always get ready to use RNA extraction kit for extracting the RNA, so that can be used to do so. And one should always standardize the amount of reagents that is used for RNA extractions. And then after extracting the RNA, we can now go forward to further downstream processes. Okay, so as you can see over here, the first is actually transcription construction of the mRNA, followed by reverse construction of the cDNA. This is followed by in silico microarray, and therefore an RNA sequencing, and therefore in silico data collection and computational analysis. So this is the entire process of transcriptomics. Now let's get to know what are other biological data that can be used in bioinformatics. The second type of data that is used in bioinformatics is proteomics. So it is the study of the complete set of proteins in a cell or system. So what is seen is that in this kind of studies, the genes provide information of our cells to make proteins, which are the machinery of the cells, and the scientists analyze any kind of tissue or cell sample to see what kind of protein is found inside it. Okay, so basically this is a large scale study of proteomes, and by proteome it is meant it is a set of proteins that is produced within an organism or any other biological context. So the proteome of a species, for example, Homo sapiens, can be a different set, and a proteome of an organ can also be a different set. So when you're talking about a proteome, it can be any proteins combinedly produced in a particular organism or a particular organ. Now, sometimes the proteome is not constant, and it differs from cell to cell and changes over time. So the degree of that like to how much extent the proteome is different reflects the underlying transcriptome, and therefore the protein activity by the reaction rate of the processes in which the protein is involved also is modulated by different factors by addition or the expression of a relevant gene. So therefore, with the help of proteomics, we can investigate when and where the proteins are expressed. It also helps us to know the rate of protein production, degradation, and the steady state abundance of the protein. It also gives us a better idea of how the proteins are modified. For example, if the protein is going through any post-translational modifications, such as phosphorylation. It also gives us an idea of the movement of the proteins between subcellular compartments and the involvement of the protein in different metabolic pathway. Further, it also helps us to know if the protein has interacted with one another. Therefore, proteomics can give us a significant amount of information of any kind of biological problems that are occurring because of a presence of certain changes inside the protein. Now it will, an example of such cases are, it can tell us which protein interact with a particular protein of interest. For example, certain tumor suppressor protein like P53 can interact with certain other kind of proteins that are present, Also, it helps us to know which proteins are localized to a subcellular compartment, for example, cell membrane. Also, it gives us an information about proteins being involved in the biological processes, for example, the circadian rhythm. So basically, if you are to see this picture, basically the area of proteomics is very wide, and proteomic experiments give us a total collective data of three properties of the proteins in a sample: the localization, the abundance, and the turnover. And also it gives us an idea of the post-translational modification. So depending upon the experimental design, the researcher can sometimes directly be interested in this kind of data, or sometimes they can use it indirectly as a form of additional information. For example, sometimes it is possible that a researcher can infer a protein's interaction with its partners, among others, with, of where it is co-localized. Okay, so depending upon how the protein is interacting with its partners, one can figure out where in the cell the protein is co-localized, or sometimes it can be assessed whether the protein is active from its post-translational modification. So basically a whole bunch of activity and interaction of the proteins can be known by the scientist by studying proteomics. Now the next type of biological data that is used in that can be used for bioinformatics studies are phenomics. Now phenomics is basically the study of the phenotypes which appear at a genome-wide scale. So it can be so basically what is a phenotype? So phenotype is describing something where that sorry. So basically, phenotype is actually describing a feature of an organism. Okay, so an example of phenotype might be a person having a certain kind of eye color, or a person exhibiting certain kind of trait. Okay, so phenotype sometimes might also be a person being at a risk of a particular disease, for example being at a risk of diabetes, or showing a certain kind of eye color. So bioinformatics uh, or studying bioinformatics give us an idea of the possible link between our DNA and a phenotype. Okay, so basically, as I said, phenomics is basically a study of the traits that make up a phenotype, and the term was first coined by a scientist in UC Berkeley known as Stephen A. Garan. And this particular area, as I said, is also interdisciplinary, and it involves research that has its place in biology, data sciences, engineering, and other fields. So phenomics is mainly concerned with the measurement of the phenotype, where a phenome is basically a set of traits, whether it can be biological or physical traits that are produced by a given organism over the course of development and response to genetic mutation and environmental influences. So the relation between phenotype and genotype enables the researcher to understand and study pleiotropy. Therefore, the concept of phenomics is often used in fundamental genomics and pharmaceutical research, metabolic engineering, agricultural research, and sometimes it is increasingly used for doing phylogenetics. Okay, so let's move on to the last category of biological data where bioinformatics can be used, that is cheminformatics. So basically it is a computational analysis of chemical and biochemical data. Herein the researcher check how a drug will interact in a organism's bodies, and after doing the experiment, that data is being stored and it's given access to all the scientists. So basically it's about the drug research, and the big databases of the drug information at different parts of the world can help scientists to develop new drugs by providing examples of the chemicals that target a certain protein. So basically, it's also a database to check how a drug interacts with a uh, certain kind of protein, and what a drug can do to a particular organism. So this is it about bioinformatics and the data that it will cater to. So as you can see that of how whatever we have discussed till now, you can understand that how much large variety and large amount of data can be uh, accessed or can be studied in bioinformatics. So with that, let's go to the next question. So the next question is a match the following question. So you have two columns, one having a set of options and another having its corresponding answers. So the first column has metabolomics, genomics, metaproteomics, microarray, and column B has complete protein analysis generates by large mass spectra libraries, expression analysis of thousands of genes at once, DNA sequence information, complete metabolite analysis. So basically all these options you have to match up within column A and column B. So if you, yeah, yeah, which one is the correct option? Yes. So can I get your name? Can I get your name? Okay, anyway, option A is the correct answer. So yes, so as you can get to know that basically metabolomics should be correctly matched with complete metabolite analysis, genomics should be matched with DNA sequence information, and metaproteomics should be matched with complete protein analysis that generates large mass spectra libraries, and microarray is basically uh, expression and analysis of thousands of genes at once. So all these are correctly matched in option 1. Now let us first check, or let us first get to know what, like briefly about what is metabolomics. So metabolomics is basically a large scale study of small molecules which are also known as metabolites that occurs within cells or any tissues or organisms. So collectively, these small molecules are basically changing at a given amount of time within cells, and this small molecules basically undergoes interaction within a biological system, and that whole array of small molecules is known as metabolome. And therefore metabolomics is a powerful approach because it actually deals with metabolites and their differ varying concentration inside the cell, and it directly reflects the underlying biochemical activity or the state of cells or tissues whose metabolic activity we are checking. Therefore, basically, it gives the best representation of a molecular phenotype. Now, when we are talking about small molecules, what are the small molecule we are exactly talking about? So basically, it is a low molecular weight organic compound that is involved in a biological process as either a substrate or a product. So metabolomics basically takes into a study small molecules with a mass range of 50 to 1500 daltons. So it can be a sugar, it can be a fatty acid, for example, it can be glucose, it can be stearic acid, it can be amino acids, it can be phenolic compounds or alkaloids. So there is as I said before also, when a cell is live, actually there is a great deal of biochemical pathways that is going inside the cell. And as a result, there is a great deal of variation in the metabolites that is produced between two species of uh, cells, and therefore it is estimated that around uh, about 20 lakhs metabolites are present in plant kingdom, whereas 7,000 and 15,000 individual plant species are present, and in humans there are around 3,000 endogenous common metabolites. And so basically these numbers give you a like a idea of how much data we will be checking when we will be checking different cells that are producing this many amount of metabolites. So basically in metabolomics, uh, we are actually taking a snapshot in time. So we are basically looking at many reaction that is taking place continuously within cells and which is resulting in variation in the concentration of metabolites inside the cell. And therefore, since we are looking at the metabolomics or the metabolites inside the cell at a given time, so basically they are very dynamic and they are changing rapidly at one point of time to the next, because certain metabolic pathways will get stopped, certain will be started, certain metabolites will be transferred from one cell to another, and therefore this particular uh, analysis is actually very dynamic. Okay, so what are the metabolic reactions that we mainly look at? We look at metabolic pathways where there is a series of chemical reactions that are catalyzed by enzymes, and the product of one reaction becomes substrate of the next reaction. So the metabolome of a cell is a complete set of metabolites within the cell, or it can be a tissue or a biological sample, depending on what sample you get at any given point of time. So it is inherently, as I said, it is inherently very dynamic. So the small molecules are continuously absorbed, synthesized, and degraded. And sometimes they are also interacting with other molecules both within and between the biological systems. So the main metabolic activities, as you can see, can be binding of one metabolic molecule with another, and also simultaneously dissociating with that metabolic molecule. It can also be degradation of one metabolic molecule into two parts or more than two parts. Sometimes it can be modification of a metabolic molecule. Sometimes it can be transport of the molecule to and fro the cell, or it can be interaction of the metabolic molecule with others uh, outside the cell or inside the cell. So basically, this is what a metabolome is. Now, why is studying metabolomics important? It is because, firstly, it is a non-invasive nature of study, and also studying metabolomics will help us to link its phenotype, and also it will help us to understand pharmaceutical, healthcare, and agricultural industries to flourish, because this particular part of study will help in biomarker discovery, drug safety screens, and there are many other studies which will help us to take our research in this field, uh, advance our research in this field. So basically, personal treatment, if you see, if we check a particular cell's reaction to a particular drug, then we can also modify the personal treatment depending on the metabolic activity of the cell in the presence of the drug. So basically, metabolomics is used on a various levels, from product and stress testing in food industries to pesticide and its identification of potentially harmful bacterial strains. It is also used in research agriculture and also for medical diagnosis in healthcare. Also, it finds its future application in personalized medicine and personalized treatment strategies. So now we have got a brief idea of metabolomics. Now let's see what is genomics. So genomics is a interdisciplinary field of biology focusing on structure, function, evolution, mapping, and editing of genomes. So a genome of an organism is basically a complete set of DNA that includes its genes and its hierarchical three-dimensional structural configuration. So if you are aware of what genetics is, genomics is actually very contrast to genetics. And in genetics, we actually study the individual genes and their roles in inheritance, whereas for genomics, we actually look at the collective characterization and quantification of all the organism's genes and their interrelations and influence on the organism. Like what kind particular gene will result in what type of influence of the phenotype on the organism. Okay, so the genes may directly lead to the production of proteins with the help of enzymes and certain messenger molecules, and we know that these proteins will be used up by the body such as organs and tissues uh, to control different kind of chemical reactions and to carry signals in between the cells. So therefore, with the help of genomics, we can study the sequencing and analysis of the genomes, and thereby we can use it for high throughput DNA sequencing bioinformatics, and thereby assemble and analyze the function and structure of the entire genome. So basically, certain advances in genomics have been done, and that has triggered the revolution in discovery-based research and systems biology, which has facilitated the understanding of the most complex biological systems such as the brain. So basically, genomics can help us to get to know about the disease susceptibility and screening and diagnosis of certain diseases. It also will give us a prognosis and therapeutic decisions of how to cater to a certain disease. Also, it will help us to know if there is any problem that a newborn baby is suffering before it is too late. So all these things can be done with the help of genomics. Now, what is the primary difference between genomics and proteomics? So genomics basically refers to the study of the entire set of genes in a genome, whereas proteomics, it refers to the study of the entire set of proteins that is produced by the cell. And therefore, in genomics, it involves mapping, sequencing, and analysis of the genomes, whereas for proteomics, it involves 3D structure, function of the proteins, and protein-protein interaction. For genomics, there are structural genomics, functional genomics, which are the two types. And for proteomics, there are structural, functional, expressional proteomics, and this correspond to the three types of the proteomics. The important areas of genomics include genome sequencing projects like Human Genome Project, where the entire genome of the human was sequenced, whereas the important areas in proteome database include development of Swiss 2D page, which are the software required for checking like I said, 2D folding of the proteins, and sometimes the software are used for computer aided drug design. So basically over here you can understand how different types of bioinformatics cater to different kind of studies. Now let's go to metaproteomics. So this is also known as community proteomics or environmental proteomics. Basically we are looking at when we say metaproteomics, we are basically looking at the experimental approaches to study all proteins in the microbial communities and microbiomes of the environmental sources. So basically, this particular field of bioinformatics is used to classify experiments that deal with proteins identified and quantified from complex microbial communities that are occurring in the environment. Metaproteomics approaches are comparable to gene-centric environmental genomics or metagenomics. So, if you check the workflow of metaproteomics, it allows the scientists to understand organisms' gene functions because from a DNA, it is transcribed into RNA and then it is translated into protein. So, when this change in gene expression is observed, then one can obviously further monitor what changes will happen in the protein due to this gene expression change. So basically the proteins that represent this particular gene will also go through a change because of the difference in expression of the gene. So metaproteomics will therefore be a tool to assess the composition of the microbial community in terms of the biomass contribution of the individual member species in the community, and also therefore it helps us to complement approaches and assess community composition based on gene copy count such as 16S RNA gene amplification and metagenome sequencing. So basically what happens in meta proteomics is that from an environment, be it your gut microbe or from environmental microbe, it is sampled and then bacterial extraction is done, followed by total protein extraction, and then it involves separation, identification, and quantification of the different kind of proteins with the help of uh, mass spectroscopy, and then it is used to design a database where a particular data is kept which will correspond to the certain kind of proteins or gene being expressed by the gut microbial organisms, or from the environment from which the organisms have been extracted. Now the third option was microarray. So what is microarray? It is basically a laboratory tool that is used to detect the expression of thousands of genes at a given time. Now DNA microarrays are basically microscopic slides in which thousands of printed tiny spots are present at a defined position. And what are these spots? As you can see over here, you can see this is basically a glass slide with sub printed spot, and each of this spot contain a known DNA sequence or a gene. Now these particular slides are known as gene chips or DNA chips. So the DNA molecules attached to each of these slides basically acts as probes and detect certain kind of gene that is being expressed in the sample. So this particular gene which is to be found out, they are known as transcriptome or a set of messenger RNA transcript. So like what are microarray used for? So in microarray analysis, mRNA molecules are basically collected from both experimental samples. So sometimes the experimental sample can be a healthy cell, or sometimes it can be a pathological cell. So basically it will be an experimental sample or it will be a reference sample. Now, depending on where the depending upon which sample, the reference sample could be collected from generally a healthy individual, and experimental sample can be collected from an individual with a disease, like for example cancer. Now the two mRNA samples are then converted into the complementary cDNA, and each of this sample is labeled with a fluorescent probe of different color. Now if you take an example, the experimental cDNA sample can be labeled as green. As you can see, it can be labeled as green fluorescent dye, and the two samples are then mixed together, and they are allowed to bind on the microarray slides. So in this process, the cDNA molecules bind to the DNA probes in the slide, and by the process of hybridization. After hybridization happens, the microarray is scanned to measure the expression of each gene printed on the slide. Now, if the expression of a particular gene is higher in the experimental sample than the reference sample, then the corresponding spot in the microarray appears red. Now, in contrast to the expression in the experimental sample is lower than the than the reference sample, then the spot of the microarray slide will appear green. So basically, if there is an equal expression, then the spot will appear yellow. So the data gathered through microarray can be used to create differences of expression between of gene expression between a healthy cell and a live cell, which will give us gene expression profile, which shows us the changes in between the genes in response to a particular condition or after a treatment with a particular drug. So basically this was all about question 2, where we discussed what are the different types of omics, that is metabolomics, metaproteomics, and we also discussed a bit about genomics. We also discussed what a microarray does, and with that we will focus into our third question, that is considering the following steps in metagenomics, what do you think is the correct sequence of order? So the first step, is it DNA extraction followed by statistical analysis followed by shotgun, then library preparation, genetic sequencing, alignment and annotation, or is it shotgun followed by library preparation, followed by genetic sequencing, followed by alignment and annotation, followed by statistical analysis, and then DNA extraction? Or it is statistical analysis, shotgun, library preparation, genetic sequencing, alignment and annotation, and then DNA extraction? Or it is first DNA extraction followed by shotgun, then library preparation, then genetic sequencing, alignment and annotation, and finally statistical analysis. So what according to you will be the correct flow of steps in metagenomics? Yes, Amandeep, its correct answer is D. Yes. So basically, metagenomic involves the following workflow, as you can see over here. First will be sample collection, which is actually not mentioned in the option. So depending on which environmental sample or biological sample you are trying to analyze, you have to collect the sample. So the sample can be collected from soil, water, it can be seawater, pond water, or any kind of dirty water, or sometimes it can be collected from biological materials like feces or urine or any other sample. And then the first step is basically DNA extraction. Any one of you has done DNA extraction in laboratory? Any one of you is familiar with the practical step of DNA extraction? Okay, what did you use to, like, what was the first step of DNA extraction? So the first thing that of that addition of, no, the first step is breaking of the cell wall, right? Or cell membrane, depending upon the kind of organism you are getting. So basically, we add lysing agent. We add agents that lyse the cell. We can actually physically lyse the cell by sonication, or we can add detergents or heats. We sometimes add SDS, sodium dotacyl sulfate. So when you do it in lab, you generally add certain quantity of SDS uh, to actually lyse the cells of the sample that you want to extract DNA from. So basically, heat is also used in certain cases, but heat is not a good method to do because it can also damage your uh, DNA or your genomic samples. So basically, certain amount of detergents and enzymes can be used for lysing the cell, and then once the DNA, once the cell is lysed, you can see that the DNA will come out into the open. So now the DNA is into the open, so we remove the contaminants and we give the DNA stability. So during this time, EDTA or NaCl is used to prevent any damage from the DNAs that can be present in the environment of the lysate. So during this process, centrifugation can also be done, and therefore that removes larger cell debris, and therefore we work only with the supernatant, which has our extracted DNA. Now, once the nucleic acid is totally extracted, it gets precipitated into a visible form, and that happens after addition of alcohol. So basically, isopropanol is used for precipitation, and after that, washing of the alcohol and other proteins give us free DNA or pure DNA. So basically, this is the first step, that is DNA extraction. Now, once you have extracted the DNA, you have to go with DNA sequencing. So basically, sequencing is basically reading the DNA or reading the way in which nucleotide sequences are present in the DNA. So sometimes you can perform Sanger sequencing. We had discussed it in our much previous classes as to how Sanger sequencing works. So previously, as an old school method, Sanger sequencing can be done, or other new platforms have come out, that is NGS, that can also be used. Sometimes shotgun sequencing can also be used, which is mainly the most accurate and widely used technique now. So basically, depending upon what platform is used for DNA sequencing, then DNA is fragmented into the smaller pieces and it is cloned into a bacterial vector, or sometimes it is directly employed for sequencing. So whatever is the process, the end result is that the sequence would be present in a file containing the nucleotide sequences, uh, and this is basically done in order to know what kind of nucleotides or what sequence of nucleotides are present in the genomic DNA. Now, once this step is done, we proceed on to binning. So basically, binning process is actually the grouping the assemblies to manifest how much two organisms are closely related. So the binning comes from sequencing two nucleotides, and once it is done with the help of computational tool, the information can be checked, and therefore two similar nucleotide sequences can be binned into one. So in during binning, we get certain unknown sequences, and therefore we compare them with a already present genetic database that we have. So, and therefore, from this comparison, we get to know how much that particular gene whose sequence we have got now is closely related to that gene. And therefore, depending on the comparison, the function of the gene is determined, and therefore, depending on that, the microbes are grouped depending on the particular function that it can perform. And this is basically all defined by the kind of sequence of the nucleotide that is present. Sometimes sequences, the binning process happens depending on the relatedness of the reference genome, and therefore to understand this binning process, sometimes it is actually important that we get to know the sequence or the genome sequence of the organism. After that, we go with gene prediction. So after binning, we actually get a rough idea about what the genome sequence will have, or from which particular category a particular bacteria is belonging to. Now, for more rigorous analysis, for example, functional analysis, further computational processing needs to be performed. And this is basically the next step comes into play, that is genetic prediction. So basically it allows prediction of the potential genes that is present in the assemblies, therefore the protein that is produced by such assemblies. And this idea will give scientists the idea about the function of the entire metagenome. Now, depending on the presence of certain types of genes and the number of the functions that is particularly this community is performing inside the system, one can think how much a particular uh, microbe is related to the other microbe. So once we get enough information about it, further processing is done. Then comes gene annotation. Now, in this process, the predicted protein sequences and the functions are matched with the protein databases bases that is available. So previously we had matched the nucleotide sequences. Now we are doing with the protein, matching it with the protein databases, and therefore it allows us to create relatedness based on the functional analysis. Now, based on each protein and its function and its comparison with different protein families, the functional classification is carried out. So this all helps us to understand the functional diversity or how much related a microbial community is in the sample. And therefore, this annotation gives us application in several processes. Okay, so basically, if we again go back to the question, we can see the first step was the DNA extraction, then shotgun sequencing, followed by library preparation, and genetic sequencing. Basically this is one step, and then this will be followed by alignment of the gene with the already prepared database and thereby annotation, and then statistical analysis depending on uh, there is how much there is a match with the particular data that we have, and therefore giving us an estimate of the relatedness between the species or between the functionalities of two species. So with that, we come to the next question. So the next question is, it is an algorithm to compare the primary biological sequence information such as amino acid sequences of proteins or nucleotides of DNA or RNA sequences. What is it? Options are BLAST, FASTA, Velvet, MacVector. Yes, the correct answer is BLAST. So have anybody of you done BLAST in any of the or checked BLAST in your computer? Yes, so if you have done it, in your left right hand side, the screen that you are seeing is what it looks like, what the main page of BLAST looks like. So if you go in Google and if you type BLAST, and if you search in the first option, this is what you will get to see. Now depending, so you can see that it is divided. If you check the web page, basically you can see two types of blast, nucleotide blast and protein blast. So this gives an idea that basically BLAST, which is the full form, is basic local alignment search tool. That is B for basic, L for local, A for alignment, S for search, and T for tool. So it is an algorithm and a program which basically compares biological information, or basically it compares sequence information. And the sequence can be amino acid sequence or nucleotide sequence, or sometimes it can compare RNA sequence also. So depending upon when you do a blast search, depending upon the results of the blast search, you can compare a subject protein or a nucleotide sequence, which is known as the query sequence, with a library or a database of sequence, and therefore identify how much resemblance or how much match is there in between your sequence and the sequence already present in the data. Now, this is how the discovery of previously unknown gene inside a mouse have been done, and if you get to see such similarity, you can actually draw the resemblance depending on the tree, phylogenetic tree. So, as I said, BLAST stands for Basic Local Alignment Search Tool. Now, it is a program which uses scoring matrices for performing sequence similarity between a variety of sequences or databases, be it nucleotide or amino acid, to give us high scoring uncapped segments among related sequences. So basically, it's like if there is a match, they will score it as 1, and therefore, depending upon the score, it will give us how much relatedness is seen between the two sequences. And so I will not go much detail into this coding mechanism because already I also don't know much about it. But basically, this step is complex and it has multiple steps and many parameters are actually looked into. So, but however, you can do this online on your system, and you can see that this algorithm is fast, accurate, and therefore, most importantly, it is web accessible. That is, that you can also do, and we can also do, sitting in our lab or home. And the relative, and it is very much faster as compared to other search tools. So depending upon the type of comparison that you have to do, the algorithm of the BLAST can be divided into the following. So the first is BLAST N. So what happens in BLAST N? It compares a DNA query sequence against a DNA database, allowing for gaps. So gaps is the part of the DNA which does not match, or the nucleotide sequence that does not match. So BLAST10 compares two nucleotide sequences or two DNA sequences. Okay, so your query also should be a DNA, and what you are searching against also will be a DNA database. Now comes BLAST-P. It compares a protein query sequence against a protein database. So basically, amino acid sequence against a amino acid database. BLAST-X compares a DNA query sequence that is translated into 6 reading frames against a protein database. Okay, so basically DNA sequence will be compared to a protein database, and in T-BLAST10 it basically compares a protein query sequence against a DNA database that is translated into 6 reading frames. In T-BLAST-X we compare DNA query sequence which is translated into 6 reading frames against a DNA database that is translated into 6 reading frames. So basically T-BLAST-X, it does not allow gaps. So as you can see, there can be multiple comparisons that can be happening. DNA can be compared to DNA. Sometimes DNA query sequence can be translated and compared to protein database, and the reverse can also happen. So this many options are there. We also have other options, that is MegaBLAST, which is used for comparison of large sets of long DNA sequences. There is also RPS BLAST, which shows conserved domain detection between different kinds of sequences that we enter. BLAST2 sequences is used for performing pairwise alignments for two chosen sequences. Genomic BLAST is used for alignments against selected human, microbial, and malarial genomes. PSI BLAST is used to construct multiple alignment from matches. PHI BLAST is used to specify a pattern that hits must match. So as I said, BLAST works on a bit complicated method, which is basically scoring depending upon the relatedness of two sequences. So the first step that the BLAST does, it first searches for short sequences of given length known as W, which is also called words or substrings, and it scores at least T, which is compared to the query sequence, and that aligned with the sequences in the database, that is the target sequence. And this leads to the formation of a matrix. Now for every pair of sequences, that is query and target, they have a word or words in common. So basically there will be a certain part of query or certain part of target sequence that are common. So then what does BLAST do? It extends the alignment in both directions to find alignments that scored greater. Sometimes it can be below a certain score threshold, that is S. Now these alignments are then called high scoring pairs or HSP. So all these are not much required. So these are basically the how this algorithm works. So if required, you can basically look up to in YouTube. You can get to see or get to understand. They will show you in videos how the scoring mechanism works. But this is not much important for understanding. So basically, if we have to summarize, BLAST is basically used for, let's go back to the question. It is used to compare primary biological sequence information that can be nucleotide or protein sequences with each other. Okay, so that is BLAST. Okay, now coming to the next question. Which of the following 16S rRNA database is highly curated? Your options are NCBI, BLAST, FASTA, QUIME or Mothur. Green genes. Anybody else wants to answer? Yes, that's the correct answer. So all these are actually nothing to do much with understanding. These are all, general knowledge stuffs. So basically, I put a picture of the of how the software tool looks like. If you search GreenGen, this is what you will get in your Windows. So basically, it's a 16S. If you look at the first line, it is basically a full-length 16S rRNA gene database, and it provides a curated taxonomy-based de novo tree interference. So this is the correct answer is basically green genes. Now, since somebody said FASTA, so let's look what FASTA does. Uh, so what is FASTA? So FASTA is basically a format. Basically, it is a text-based format for representing nucleotide sequences uh, or amino acid sequences. So in this particular tool, nucleotide or amino acid sequences are represented using single letter codes. So you are aware that proteins also are represented with single letter codes like nucleotides. So what this format does is that they align all the different types of proteins and DNA, and therefore they allow. Yes, anybody wants to say something? Okay, sorry. So basically, it allows to search for similarity between the protein sequences or the nucleotide sequences. And the simplicity of this format helps us to manipulate and put sequences depending upon the scripting language. So basically, the uses are actually that it can be used to align the protein and the DNA sequences, and the translation and algorithm helps us to handle frame shift errors. So if there is a error in while there is a shift of the transcription frame, that also can be done in FASTA. So we also can calculate the similarity and therefore this between two sequences, and therefore this will help the scientist to decide if the alignment of the nucleotide has occurred correctly, or it can help us to know on about the homology, or it can help us to infer about the homology between the two sequences, or the amount of relatedness between the two sequences. So like BLAST, FASTA also have programs. So FASTA programs can be listed as follows. So first is FASTA A. So it is basically a protein-protein sequence comparison or a nucleotide-nucleotide sequence comparison. FAST X or FAST Y is basically nucleotide protein sequence comparison. S search program in FASTA is for local alignment between the protein-protein or nucleotide-nucleotide sequence. GG search actually allows us for global alignment between protein-protein or nucleotide-nucleotide sequence. GL search is the program that helps us to allow global alignment of the query and local alignment of the sequences in the databases. So all these FASTA programs you can use in the window and you can get the required results. Now, since we got to know about BLAST and FASTA, let's check what is the basic difference between BLAST and FASTA. So BLAST, as we said, is an algorithm for comparing primary biological sequence information like nucleotide or amino acid sequences, whereas FASTA is a DNA and a protein sequence alignment software, and it is hosted by a European Bioinformatics Institute. So it is BLAST is mainly for comparison, whereas FASTA is mainly for alignment. So it aligns and therefore helps us to know about frame shift. So what does BLAST use? It basically uses local sequence alignment. FASTA also uses local sequence alignment, and then sometimes it can extend to global alignment, wherein the entire sequence or entire big sequence of the two nucleotides or two proteins are aligned. Now in BLAST, it searches similarities in local alignment by comparing individual residues in the two sequences, whereas for FASTA, it also searches for similarities in local alignments by comparing sequences or patterns or words. So this is how the two uh, operate. And then in BLAST, for more similarity or for better similarity, searching is done or it is closely matched for optimal sequences or locally optimal sequences, whereas for FASTA, for better similarity, searching is less done in less similar sequences. So BLAST is seen to work best for protein searches, whereas FASTA is seen to work best for nucleotide searches. Now, even though BLAST is a sensitive tool for bioinformatics, FASTA is known to be a much more sensitive tool as compared to BLAST. In BLAST, we can see there is gaps between query and target sequences are not allowed, whereas for FASTA, these gaps are allowed. Uh, BLAST is seen to be much more faster than FASTA, whereas FASTA is comparatively slower. So all these are examples. So for uh, getting a much more idea about all this bioinformatics tool, you can go ahead and run a particular sequence of your choice, and you can then compare it with a sequence in the total or the entire data sequence that's there, and you can let me know how much there was similarity in BLAST and how much there was alignment similarity in FASTA. So with that, we come to end of the live interactive session. So I guess all the sessions have been addressed, and if anybody wants to ask questions, you can ask. Otherwise, we can end the session. Is there any questions? In that case, we can end the session for today. Thank you for your tension.